In this presentation, I will give 4 short case studies to illustrate the power of this technology that can solve many problems you may meet in food industry. First case: which microorganisms are causing alteration of my product? In this example, a meat producer had a serious problem with a fermented meat product. It was salami. Several production batches were lost with non-conform products. However, all the results with classical microbiology were conform. The producer suspected that the ferment was the cause of the problem. Quality partners suggested the use of metagenomics to identify the origin of the contamination. 3 samples were analyzed: the ferment, the conform product, and the non-conform product. In this graph, you can see the proportion of the different bacteria in the 3 samples. The bacteria in the ferment corresponded to the technical sheet given by the ferment producer, so the ferment was conform. The conform meat product contained only bacteria from the ferment, but the non-conform meat product contained bacteria that were not present in the ferment. Lactobacillus brevis is described in the literature as a bacteria from the environment that could spoil the meat. In this second example, what is the evolution of the microbial flora during conservation? For this study, we follow the evolution of bacterial population of minced meat under different packaging conditions, under food wrap and modified packaging. We took 3 samples, firstly at the beginning of shelf life, secondly at the end of shelf life under food wrap, and thirdly at the end of the shelf life under modified atmosphere packaging. In this graph, you can see the identification of bacterial species and their concentration in log CFU. The sample at day 0 is represented in blue, under food wrap red, and under modified packaging in green. Pseudomonas is mainly present at high concentration under food wrap. This bacterial genus is well known for spoiling meat products. Lactic acid bacteria is mainly present under modified atmosphere packaging. Some of them could spoil or protect meat products. Brocotrix thermosphaca and leuconostoc are present in both conditions. In conclusion, with metagenomics, we can follow the evolution of bacterial population according to environmental conditions. With this technology, we have a clear picture of good and bad microorganisms present in food products. In this third example, how can I increase the shelf life of my product? A meat producer wanted to extend the shelf life to export to foreign countries. This product was head cheese, a terrine made with flesh from the head of a calf or pig. Quality partners suggested the use of metagenomics to validate the effect of lactate diacetate solution to control bacterial flora in this meat product and so to extend its shelf life. This graph gives the proportions of bacterial species. On the left, with lactate diacetate, on the right, without lactate diacetate. Spoilage bacteria such as Bronchotrix ceracea, Pseudomonas, were identified in the product without lactate diacetate, while Carnobacterium is mainly present in the product with lactate diacetate. Some Carnobacterium species are described as protective flora for meat products. Thanks to a mix of lactate and diacetate, Carnobacterium dominates the total flora and has a protective effect on the meat product. In conclusion, metagenomics validates that adding a mix of lactate and diacetate to the head cheese increases the shelf life. In the fourth example, what impact will a change in recipe or packaging have? In order to control the quality of raw meat from different producers, Quality Partner analyzed the bacterial flora of 6 tartare steaks. Steak tartare is a dish prepared with raw minced beef meat, spices, and vegetables. This graph gives the proportion of bacterial species in 2 restaurants, 2 sandwich bars, 2 butchers. At 2 butchers, 1 bacterial species was mainly present, Lactobacillus algidus. This bacteria is present on fresh cut meat, and the presence of this bacteria is an indicator of freshness of the raw beef mincemeat. In the sandwich bars, a much higher bacterial diversity was noticed. This diversity was due to the bacteria from vegetables added in these products. In the first restaurant, the quality was still good, with a high proportion of Lactobacillus algidus. But in the second restaurant, spoilage bacteria was present in high proportions, and the product was completely spoiled. This graph gives the concentration of some bacterial species in the samples. The bacterial concentrations were low for 2 sandwich bars and for the first butcher. The concentration was higher in the second butcher, but it was mainly composed of Lactobacillus algidus. In the restaurants, the concentrations were higher, but it was especially in the second restaurant that the spoilage bacteria was present in high concentrations. This case study shows the power of this technology to identify and quantify bacterial flora. High concentration is not always correlated with spoilage. Some bacterial species could be present in high concentration, but the meat product is still of a good quality.